A very big hello to my future champions out there. I hope you're doing absolutely good, and I also hope that you have watched the one-shot lecture of Principles of Inheritance and Variation. My dear children, I guarantee you, if you watch the 3 videos that have been posted today, you are going to get full on full in this chapter. And my aim is not midterm, my aim is boards. I am pretty sure if you're watching these videos, it won't be just useful for your midterm but also for your boards. I can guarantee you 13 on 13 in this chapter. And if you have that trust on me, put it down in the comment section that yes ma'am, we trust you. And why are we covering this session today? What is the use of this one marker session? Because according to the previous year blueprint, my dear children, you got at least one marker question from this particular chapter, that's Principles of Inheritance and Variation. And the total marks of this chapter is what? 13. So ma'am, only for one marker I am going to see the whole lecture? Yes, not just for that, but also it would help you understand the concepts. And these concepts are something that would be repeatedly going to be asked in your 2 marker as well as your 5 marker. So it's like a revision that you would definitely need to have before your examination. So please watch it. And I have given you one homework question towards the end of the session. If at all, you have understood each and every bit of it, put down the answer for that in the comment section below, and I want you guys to tell me how is this video for you guys. Is it helping you well or it is not? Was it really good or not? Okay, and as I said, if you trust me, you have to put it down in the comment section. We trust you, JJ ma'am. Okay, the first question that we have is this. 37.2% recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with phenotypes red eye color, normal body, white eye color and miniature body is due to. So many things have been given, so many characteristics have been given, but what do we have to focus? We have to focus on this part of the question. 37.2% recombinant. That means you're talking about the percentage of recombination. What is recombination? The formation of new things, right? Recombination. There are new things being formed out there. So what generally happens over here, my dear children, is that you try to find the answer. If you know the answer, tell me. Okay, anyway, I'm explaining for those of you who do not know. So over here, suppose I have a gene A and B. Over here, I have a gene capital A and capital B. Suppose in chromosomes, when they lie together, no, there is chances of this part to come this side and the other part to come that side. But whatever happens, if you see, small a would always remain with small b, big A would always remain with big B. Why? Because they are very close to each other. They are very close to each other, and this is what I called tightly linked genes. They are what? Tightly linked genes. Now when they are tightly linked, do you think there is new combinations being formed? No ma'am, the same combination is over here. There is very less chances that you find new recombinations out there. So the percentage of recombination over here would be what? Very less. Very less. Okay. But on the other hand, if you think of a scenario where it's something like this. Small a over here, small b over here, capital A over here, capital B over here. Now suppose this and this get swapped. The upper part gets swapped. What will happen? Now capital A will be with small b. Okay, because this got swapped, and small a will be with capital B. My dear children, you tell me, don't you think a new combination is being formed? So the percentage of recombination would be what? More. When does more recombination happen? When the genes which are there, they are not tightly linked. Instead, they are what? They are loosely linked. And what do you mean by loosely linked? Loosely linked genes means they are very far away from each other. They are not close. They would be very far from each other. So 37.2% is a huge amount of recombination. You know what? 50% is the highest amount of recombination. Out of that, if we are saying 37.2% recombinants are being formed, that means the genes which he have been talking about, my dear children, they are loosely linked first of all, and if they are loosely linked, they would be far away from each other. So loosely linked and short distance between the genes, wrong. It is long distance between the genes. So loosely linked and longer distance between the genes becomes your correct answer. My dear children, always understand the questions that I am dealing over here are from your previous year question bank, are from your model question paper, are from your previous year question paper itself. So make sure you are practicing with me, because as I said, if you practice these questions with me, I can tell you, you're getting 13 on 13. Okay, so going ahead, I hope this particular thing is clear. and with every answer slide, I will be giving you from which part of the book it has come. So it belongs to what? Linkage and recombination topic. You will see so many questions from this topic. Now going ahead, you have the next question. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. Dihybrid cross means you're talking about two characters. For example, the color, that is yellow and green, and color of the seed, and also the shape of the seed, that is round and wrinkled. So two characteristics are being studied together at once. Now the ratio over here is what? 9:3:3:1. What does that denote? What is 9:3:3:1? So when he crossed, my dear children, he found that he started with round and yellow parents, as in a plant that had round seeds and yellow seeds, and also he crossed it with wrinkled and green seeds. These ones are the dominant character, this is a recessive character. So there was a parent with dominant character, there was a parent with recessive character. When they crossed, he found that all the first generation were round and yellow because the dominant character is the one that expresses in the first generation. In the next generation, he found something very unique. It was not just round and yellow or wrinkled and green, there were new combinations being found out there. So 9 of them were the dominant ones, that means you're talking about round and yellow. One of them was the recessive one, that means you're talking about what? Wrinkled and green. And over here you found new combinations. So since round was with yellow, we make the new combination, that is round with green this time. And over here wrinkled is with green, right? So we'll make a new combination, wrinkled with yellow this time. So 6 out of all of them were what? The new combinations that he found. But how come you have newer combination? That is the question. Why do we have 9:3:3:1 ratio? Why it's not just round and yellow and wrinkled and green? That's the question. So why is that? Because the alleles of two genes are segregating independently. Didn't understand? I'll tell you. So over here, suppose this is for capital R for round and this is for small, that is what you call wrinkled, and over here it is for yellow and this is for green. Every parent will have a chromosome pair. Out of these chromosome pair, only one chromosome would be coming in a gamete from one parent. Similarly, from another pair, only one would be coming from a gamete. Now from this pair, either capital R can go or what? Or small r can go, right? Suppose capital R came from here, okay, and from this point you got capital Y. So the gametes are what? Capital R, capital Y. Suppose capital R came from here and from this pair small y came. So you got the next type of gamete. Now think, over here you have the small r coming and then from here you have the capital Y coming. This is the third type of gamete. Now small r and then small y coming. This is the fourth type of gametes. So what do you understand from that? The alleles, these are alleles of one gene, this is one gene, and this is the second gene coding for the second character. This is the second gene coding for second character. It is not like capital R will only go with capital Y. It is independent. From this pair anyone can come, from this pair anyone can come. And when you have newer gametes, these gametes are gonna come from here. Suppose you have got capital R, capital Y, from there you got small r, small y. These gametes are gonna come and form something new out there. That is how you get newer combination. You get it? They are independently assorting, and this is what the third law of Mendel is, law of independent assortment. When you're talking about alleles of two genes coding for two characters, they are going to segregate independently. Okay, so your answer would be D, not A. The alleles of two genes are interacting with each other. If they are interacting, then it should be no new character should be seen. It is a polygenic inheritance. Polygenic inheritance means multiple genes are responsible for one character. No, here one gene is responsible for one character, that is the shape. Another gene is responsible for another character, that is the color. Now it is a multiple allelic inheritance. No, you do not have multiple alleles. One gene has two alleles, that is normal. Here also one gene has two alleles, capital Y, small y, that is also normal. So your answer would be D. Good? Done? Chalo. So this is what law of independent assortment, and my answer is D. Now children, we would be going a bit faster because if too much of things are explained, it's of no use because I've already explained in the one shot revision video. Watch it. Now a pedigree chart is given below. Identify the trait responsible for this inheritance pattern. So my dear children, this is the pedigree chart. These are the options. Whenever remember this golden rule. Whenever you do not see a diseased, diseased organism or a human being in all the generation, that means it is a recessive trait. If you see that over here this is colored and then this is also colored, and then if at all this guy has some child that is also colored, then if in every generation you see a child who is being diseased, that means that is a dominant trait. But over here in the first generation itself, you see that no one is being diseased. No one is diseased, which is why it is not colored. So this is what? This is a recessive trait, that is for sure. So I am writing recessive trait. If it was dominant, it would be in every single generation. That is, this parent, at least one parent would be diseased. Here also one parent should be diseased, and that's how it goes. Now, is it autosomal or is it sex-linked? What do you think so? Is it autosomal or is it sex-linked? So over here, my dear children, the boy is affected. Here the girl is affected. Here the boy is affected. But you clearly see it's not just the boys who are affected. Do we see that? No. We do not see that, and with that we get an idea that it would be what? A autosomal. It is not sex linked. It would be what? An autosomal recessive disorder, or to the trait out there. So it is what? Autosomal recessive trait. Fine? Cool? Chalo. If it was sex linked recessive, it would be the males who are mostly affected. Okay, please remember, if it is a sex linked disorder, it would be the males who would be mostly affected. Cool. Let's go. Which of the following is not a recessive gene linked disorder? Which of them is not a recessive gene disorder? That is what is being asked. What do you mean by recessive gene disorder? That means, for example, suppose there is a chromosome. Okay, so over here, if suppose this is the condition where there is a normal X and there is an X which is affected, will the person will have the disease? No, because it's a recessive disorder, and recessive disorder will appear only if both the chromosomes are affected. That's the only possibility of it to happen. Or if it is not XX, let it be something else. For example, HB. Okay, suppose HBS. This is one which is affected and this is the one which is not affected. Okay, so is it going to show the disease? Is this particular person going to show the disease? No. Why? Because recessive disorders can express only when both the alleles have this problem. Okay, it should be homozygous condition. So my dear children, over here you are being asked which is not a recessive gene linked disorder. So sickle cell anemia is a recessive gene linked disorder, hemophilia is also, and color blindness is also one such thing. Hemophilia and color blindness, as we have already studied, they are X linked recessive disorder. They are sex linked recessive disorder. Sickle cell anemia is what? Autosomal recessive disorder. Okay, but they are all recessive. Only one thing that you are left with is myotonic dystrophy. It is actually autosomal dominant disorder. So this is given in your NCERT page number 73. If you see this diagram, over here they have given a pedigree chart, and under that they have mentioned that this is an example of myotonic dystrophy, which is an autosomal dominant disorder. You do not see it in the explanation part, so do not miss this. Okay, cool. My dear children, are you liking this video? Do you think I am explaining it to you well? If at all I am, what do you have to do? Click on the like button. Let's be sure that we can go up to like 50 likes. Can we do that? Share it with your friends also. Let them also benefit from it, and make sure you are studying very seriously. Okay. Now, a plant having the genotype capital A small a, capital B small b will produce dash kind of gametes. Now, this is not directly from your NCERT. Okay, but this type of questions are there. Okay, so how do you find that if a plant has a genotype like this, how many gametes are being produced? You have to use a formula that is 2 raised to the power n, where n is what? The number of heterozygotes. Heterozygotes. So over here, capital A small a is one such heterozygote. Why heterozygote? Because this condition would be known as homozygote. Over here it is like this. That is why the two alleles are different. Similarly, capital B small b, that is also another heterozygote. So over here, what do you replace it with? What do you replace n with? You will replace n with 2. So 2 raised to the power 2 is what, my dear children? It would be 4. So your answer would be 4. 4 different type of gametes are going to be produced by them. Now depending on whatever it is, for example, I'll give you homework right away. You tell me. Suppose it is like this. Okay, this is capital C and capital C. What are the number of gametes going to be produced over here? This is your homework number 1 question. Okay, use this formula and try to find out. You have to spot the number of heterozygotes first. Okay, now, yes, so it's 4 and going ahead. 6th question, which of the following is not a pair of opposing characteristics Mendel studied? Which is not something that Mendel studied. Axial and terminal flowers, it is something. So axial is the one that is dominant. Axial means from the point where the branch comes off, a flower comes off. Okay, that is what you mean by axial. Terminal means at the tip it will be there. So axial is something that is dominant. Full and constricted pots, the pots in which, what do you call, you get the peas, my dear children. It can be full, it can be inflated, or it can be dry and constricted. So full is the one that is dominant. Green and yellow pots, which one is dominant? It is the green one. It's suppose this color is being asked. So green is the one that is being dominant. If you're asked about the seeds, it is yellow one which is dominant, not green. Remember, there's a difference. Now pink and white flowers is something that he never studied. It was violet and white flowers, and violet is the one that is dominant. So this is the odd one out. Okay, so your answer would be D. Here are the characteristics. How many characteristics are there? 7 characteristics are there, and if you talk about each character, they have 2 opposing things. So 7 into 2, how many traits? 14 traits are there and 7 characters are there. This is a character, seed shape. Trait is what? Round and what do you call? Wrinkled. That's the trait. That is why you say 14 traits and 7 characteristics. Remember all of it, very important. Okay. Now, in a monohybrid cross between 2 heterozygous individuals, percentage of pure homozygous individuals obtained in F1 will be what? In a monohybrid cross, 2 heterozygous individuals are there. 2 heterozygous, suppose, that means it should not, allele should not be the same. Capital T, small t, capital T, small t. Okay. Percentage of pure homozygous. We have to find pure homozygous. So let's cross it. This and this you get capital T, capital T. This is one homozygous. Okay, this and this capital T, small t. This is heterozygous. Alleles are not same. This and this you get capital T, small t. Again heterozygous. This and this small t, small t. This is homozygous. So out of 4, 2 of them are homozygous. This and this. Out of 4, 2 means 50%. Can I say 50% of them? So your answer would be 50. You're asked about F1 generation. Please check. In the question it can be F2 also. Okay, so your answer is 50%. Going ahead with the 8th question. In the F2 generation of Mendelian dihybrid cross, the number of phenotypes and the genotypes are. In the dihybrid cross that we did, we know the number of phenotypes. Phenotypes means physical appearance. We studied round and yellow, and then round and green, the new character, and then wrinkled and yellow, and then wrinkled and green. 4 phenotypes are possible. What is the number of genotypes? Ma'am, 9:3:1 ratio, if you calculate all of them, 16 plants are being formed. My dear children, these 16 plants do not have different genotypes. Some genotypes are repeated as well. You get it? Some genotypes are definitely repeated. So what are the unique genotypes that you see which are not repeated? There are 9 of them. 9 such genotypes. Genotypes mean genetic makeup that is not being repeated. Okay, so if you see the whole of the thing, here you see capital R, capital R, capital Y, capital Y. Okay, that is there. Agreed. Over here you see capital R, small r, capital Y, small capital Y. So this is one genotype. This is the same genotype being repeated. So I cannot count it twice. You have to count it only once. Oh my God, I'm so sorry. I didn't zoom it in. Okay. So this is there. It cannot be counted twice. If you're asked about the number of genotypes, it would be counted only once. You get it? So over here, these are the 9 unique genotypes which are there. You have to remember 9 genotypes. Do not go and write 16. 16 plants are being formed. That is there. But unique, if you're talking about unique genotypes, 9 of them. It is repeated again and again. Okay. Okay. I hope you won't get this wrong. And now going ahead. Okay. Mendel's work was rediscovered by 3 scientists in the year. What was the year? Mendel's work was rediscovered by 3 scientists in the year 1900. Always remember it. And also remember what were those years where he did his experiment. Mendel's experiment was done in which year? From which year to which year? This is your homework question number 2. I had taught you several times. You need to answer me. It has something to do with 1850s. Okay, I'm not writing the number over here, and it goes up to 18/6-. Okay, so 5- and 6-. For 7 years he did his experiment. Now, after so many years, his work was rediscovered in the year 1900, not 1910 or something like that. Okay, and now the next question comes, that is okay. The next question comes, which three scientists independently rediscovered Mendel's work? Who are the one who rediscovered in the year 1900? Tell me. It's an easy question. Now, there is no need to explain all over it. De Vries, Correns and Schemark. Okay, it's a D option. De Vries, Correns and Schemark were the people who rediscovered Mendel's work. So your answer would be D. Okay, cool, works. Chalo. Don't forget them. Okay, don't get confused between Sutton and Boveri. No. After they rediscovered Mendel's work, Sutton and Boveri came into the picture and gave the chromosomal theory of inheritance. It was Sutton who is mainly responsible for chromosomal theory of inheritance, telling you that as well. Now, going ahead, in Antirrhinum or snapdragon, phenotypic ratio in F2 generation for the inheritance of flower color would be. In F2 generation, what is the phenotypic ratio in snapdragon? If you remember, there was a red flower crossed with white flowers, and all the crosses were capital R, small r, but instead of showing red color, all of them were pink. This is what your F1 generation. Now for F2 generation, what do? You do selfing. You take capital R, small r, and you cross them. When you cross them, okay, what would be the phenotypic ratio that is being asked? According to the physical character, what would be the ratio? So over here I have already made it. It is 1:2:1. How do you get it, ma'am? So capital R, small r, if you cross this and this, you get capital R, this is red, okay, because there is no other allele. This and this goes, this would be what? Pink in color. Why pink? Because it is incompletely dominant, not completely dominant. This is an example of incomplete dominance. Now this and this comes together, again capital R, small r, again pink, my dear children. This and this comes together, small r, it would be white. So one of them would be red, okay, two of them would be pink, and one of them would be white. So 1:2:1 is the phenotypic ratio. What is the genotypic ratio? Remains the same. One of them capital R, two of them capital R, small r, and one of them small r. So phenotypic and genotypic ratio in Snapdragon is the same thing. Okay, and they show incomplete dominance. I hope that's clear. Cool. Let's go ahead. Now, genes located very close to one another on the same chromosome tend to be transmitted together and are known. Genes which are very close to each other, they are transmitted together. So what does it show? What do you call them? I already told you. First question, remember? Is it allelomorphs, identical genes, recessive genes? No. It would be linked genes. So many terms might be new to you. You don't need to be concerned about them as well. Okay, it would be what? Linked genes. And again, this is coming from your linkage and recombination. Now, 13th question, chromosome maps or genetic maps were first prepared by. What do you mean by that? Genetic maps means, okay, ma'am, this chromosome, this allele is over here and the other allele is over here. This is gene mapping. So, who discovered gene mapping? Who was that person? Ma'am, Morgan, T.H. Morgan did the experiment for linkage and recombination. Okay, he was the person who said that if there is more distance, there is more recombination and this and that. Okay, he definitely said if there is more distance, there will be more recombination. Linkage and recombination was actually put down by Morgan. But who used this information to produce something that is known as gene mapping? Where that person said that, okay, with information of Morgan, I get to know that this gene is located over here or this allele is located over here. Who was that person? It was the, what you call, student of T.H. Morgan, and it was Alfred Sturtevant. He is responsible for gene map or genetic map. Please remember that. So, D is your answer for this question. Now, going ahead. Alfred Sturtevant, don't forget him, okay? Now, which of the following... Will you also be some students like that who will be taught from me and will produce something else later on? I hope you do. Now, which of the following is incorrect regarding ZW-ZZ type of sex determination? ZW-ZZ, where do you see it? You see it in birds, first of all. Okay, you'd see it in birds. My dear children, in birds are the only case where you find female heterogamity. What do you mean by that, ma'am? Heterogamity means, hetero means, you know, different. So over here, usually we see in XX, XY in human beings, females anyhow will be producing gametes that is having X chromosome because they do not have anything else to give. But over here, they have ZW. So either they can give Z or they can give W in the gametes. So this is known as female heterogamety. Heterogamety means two different type of gametes are being produced by the females. So it occurs in birds, correct ma'am. Females are homogamitic, wrong, no. Are they homogamitic? No. And males are heterogamitic, again wrong. Males are what? Homogamitic over here. So females are heterogamitic and males are homogamitic. This is correct. Total number of chromosome is same in both the male and female. Yes, it would be same. So over here, this gamete can, this chromosome can come or this chromosome can't come. Similarly, in the case of males, either this or this. Anyway, one chromosome is being passed down. So the number of chromosomes remains to be the same itself. Okay, so over here, what is the odd one out? Which is the incorrect one? This is the one. Okay. This is ZZZW type of sex determination. Always remember, it's the only condition where females are heterogamitic. Now going ahead, a couple has 3 daughters. What is the possibility of them having a son next time? So couple, we see mother and father had 3 daughters. Can you predict the other one will be a daughter or a son? Can you do that? No one can do that because there is 50% of chance. Why ma'am? Because if you see ma'am, I was calling you guys also ma'am. Okay, it's okay. So XC, XY. So over here, this and this comes together, you get XX, that is a female. This and this comes together, XY, that is what? A male. Agreed? This and this comes together, again you get female. This and this comes together, you will be getting male. So you tell me, 50% chance of male and 50% chance of male. Can you guess what will be the next child? No. What is the possibility of them having a son next time? 50%. What is the possibility of them having a female daughter next time? 50%. So your answer would be C. Cool? Works? Thumbs up? Yeah? Okay. Next, 16. Haplodiploid sex determination is found in haplodiploid. What do you mean by that? Haploid means what? You are having half the number of chromosome, n number of chromosome. Suppose 46 is the total number of chromosome. If I just have 23 of them, then I will call it haploid. Diploid means you are having 2n. Double, that means I have 46, not half, complete. 46 would be there. So n and 2n is right over here. Now my dear children, what does it have to do with sex determination? So there is a condition and that is seen in what? Honeybees. It is seen in honeybees where the haploid ones, okay, the ones that have half number of chromosome, that would be the males, and the diploid ones, that would be females. Ma'am, how is that possible? That might be the next question. We anyway studied it, but I can explain it to you once more. I don't mind it. So over here, see, come on Zoom, yeah. So females, my dear children, I told you they are diploid in nature. So 2n, that is here, that is 32, not 46. 46 is in the case of human beings. So they have 32 number of chromosome. Now, whenever gametes are being formed, they undergo meiosis. And in meiosis, what happens? The chromosome divides into half. Okay, so half number of chromosomes, this is reductional division. So there will be half number of chromosomes going up there. So in one gamete, one half, another gamete, another half. So gametes are always haploid in nature over here. Now, if you talk about the males, they are not diploid. They are starting from haploid point itself. They have half the number of chromosomes. So, can they undergo reductional division again? No. They will undergo mitosis. Mitosis is equational division, where they will just divide into 2, but the number of chromosomes remains the same. So, they were haploid, their gametes are going to be haploid itself. Now, from the male and the female, N and N came together. What did you get? 2N. Now, whenever 2N is being formed, it will be what? It is diploid and it will be what? Female. Ma'am, suppose this N is not getting fertilized itself. This gamete which had haploid number of chromosome did not get fertilized. What will happen? It will automatically form what? Males. In the case of honeybee society, my dear children, these males are known as drones and they are just there for sexual interaction, for making babies, nothing else. Okay, so these drones are being formed, they have haploid number of chromosome. So if you have half the number of chromosome, they would be males. If you have diploid number of chromosome, they would be what? Females. So this is haplodiploid sex determination which is shown in honeybees. Okay, grasshopper? No, grasshopper is an insect, honeybee is also coming under that, but both of them are showing different things. Please remember that. Honeybees exclusively show haplodiploid sex determination. And there's a dialogue in your NCERT. Please go through it once. That says that, what is it? the fathers are there. Okay, the honeybees, they have fathers, sorry, they have grandfathers, no, not fathers. So please go through that. I have explained it once. If you want to explanation for that again, you can ask me in the comment section. I can do that. Okay, now going ahead. Now the next question, in XO type of sex determination, what happens? What is XO type of sex determination? It is found in insects like grasshopper. Okay, and over here the females are going to produce one type of gametes only, all of them having X chromosome in them. But in the case of males, they have XO condition. That means they can produce two type of gametes. One that carries X chromosome and the other which does not carry anything, which is why it is denoted as 0. Okay, it is denoted as O, as in nothing is there. So suppose this and this comes together, you get XX. Again, XX is what? Female is being going to produce. Over here, if this and this comes together, my dear children, you're getting a gamete without anything out there. So this would be what? Males. This is found in what? Grasshopper. Okay, this is found in grasshopper. And yeah, so females produce two different type of gametes. Do they produce two different type of gametes out there? What do you think so? No, it's the same type of gametes, right? Males produce two different type of gametes. Yes, ma'am. X and something that does not have anything, right? One gamete that does not have anything. So this is the correct answer. Females produce gametes with Y chromosome. No, Baba, there is no Y chromosome. Males produce gametes with Y chromosome. No, there is no Y chromosome up over here. So XO type of sex determination is found in grasshoppers. So we actually saw so many type of sex determination. ZW found in female birds, okay, where they show female heterogamety. Then we saw haplodiploid found in honeybees. Then we are seeing XO type of sex determination, that is example of male heterogamety. It's found in grasshopper. And now you have XXXY, that is found in human beings. That is also an example of male heterogamety. XXXY, this type of sex determination is also found in Drosophila melanogaster. Remember it. Along with humans, Drosophila melanogaster. Okay? Cool. Now, when is it possible for a woman to be color blind? Color blindness is a disease which is recessive disorder. That means you should be having two alleles which are affected, then only this particular disease would be coming in. So if a woman need to have color blindness, my dear children, women are like this XX condition. So both the chromosomes should be affected for her to be colorblind. If only one is affected, that means she is going to be a carrier out there. So over here, for this condition to happen, she should get one affected X chromosome from her mother and one affected X chromosome from her father. So the father would be like this and the mother can be like this or she can be like this also. But at least one chromosome should be affected. That means the mother can be a carrier or can be colorblind, but the father anyway has to be a colorblind father. Over here it would be expressed. It would be expressed even it is having one X chromosome that is affected. Why? Because there is no other X chromosome to overshadow the affected. Which is why the males are the one who are usually affected in sex-linked recessive disorders. Okay, whenever it is sex-linked recessive disorders such as hemophilia or color blindness, the males are affected. Why? Because they do not have a counter X to overshadow the effect. So even in this condition, they will be showing the effect and this guy would be what? Colorblind. So when it is possible for a woman to be colorblind, the father has normal vision. Do you think he has? No. Wrong. The father has normal vision, again wrong. The father is colorblind, correct. And the mother has a normal vision. Do you think mother can have a normal vision, my dear children? Either, yeah, she can have a normal vision, but she need to be a carrier at least. This particular person would be having normal vision, but she is a carrier. Normal can mean this also. So saying entirely like this would be wrong. This and this shows normal character. They are not colorblind. One is carrier, another one is totally normal. But since two of them are there, we cannot go with this particular statement, that is normal vision. Over here there is something more specific. The father is colorblind and the mother is a carrier. Yeah, that would be very much correct. Okay, so your answer would be D. Fine. So you have to look for options and only answer the question. Do not be in a haste to answer it. Fine. Chalo. Children, are we hitting the 50 likes yet or not? Okay, a man marries a woman and both do not show any apparent traits of inherited disease. Okay, man is there, woman is there, both of them are not showing any signs of any disease out there. Now 5 sons are there and 2 daughters are born. 3 of the sons suffer from a disease. However, none of the daughters are basically affected. How can that happen? So the father and the mother, both of them do not have any disease. 5 boys are being produced. Out of them 3 are diseased, my dear children, and 2 children, 2 daughters, right? 2 daughters are being born and they are perfectly fine as well. So you can clearly see it's the males who are getting affected, which means there are chances that this disease is what? Sex linked. This disease, there's a chance that it can be sex linked. Now, how do we understand whether it is recessive or dominant? If it was a dominant disorder, then the parents also, if they have that gene, they should be expressing it. Probably it is a recessive disorder, which is why the parents are also not expressing it. Ma'am, you told if it is sex linked, the males would anyway express it because the condition would be like this, no? But over here, the chance can be that the male is perfectly fine, the father is perfectly fine. Is the female probably who could be a carrier, which is why she is also not showing the effect. It's a recessive disorder. Now what happens, my dear children? Suppose this and this chromosomes comes over here, you get XCY condition. Don't you get a male who is getting affected? You are getting a male who is getting affected. The females, do you think they will get affected? More of a no, they will all be carriers. Okay, since the male is not affected, the females are also not going to be affected over here. So how did you understand whether it was sex linked? By understanding the ratio. Out of 5, 3 of the sons were affected, but 2 daughters were there, they were not affected. So you got a clear, idea that is, it could be sex linked. Now how do you understand whether it's dominant or recessive? If it was dominant, if the parents are unaffected, my dear children, that cannot be possible. In dominant, at least one parent would be definitely affected. If it was a dominant disorder, suppose this gene was something related to dominant disorder, then if she has even one, it would show the effect. She would be diseased, but over here she was not. That means it is what? A recessive condition, and the father is completely normal. So your answer would be what? Sex-linked recessive disorder A. Cool? Done? Chalo. Next. In sickle cell anemia, glutamic acid is replaced by, in sickle cell anemia, my dear children, the 6th codon. Okay, the 6th codon is affected. As in, my dear children, if you have mRNA, there is an mRNA being formed because of the DNA, because of the information of the DNA. Now, they have certain codons over here. They have one codon. One codon consists of 3 nucleotide bases. So, second codon, third codon, such that you have 6th codon. Over here, initially you had GAG, but this A is replaced by U. So GAG codes for an amino acid, that is glutamic acid, but now it is coding for an amino acid, that is what? Valine. So always remember, A comes before U, so A was the first one, and now it is replaced by U. Similarly, glutamic acid comes first and then comes valine. G comes first and then comes V. You can imagine like that. So in sickle cell anemia, glutamic acid is replaced by what? Valine. Your answer is A. Cool? Done? None of the other amino acids. It went there. Okay. Chalo. You come here, Baba. I cannot move like that. Okay. If both parents are unaffected carriers of thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? My dear children, here they have clearly mentioned, they have given you a hint also, that it is what? Autosomal recessive disorder. That means it can happen. It is not a problem that is linked with your sex chromosome. You have 23 pairs of chromosome. Out of that, 22 of them are autosomes and just one pair is the sex chromosome. So my dear children, I am saying it is recessive disorder. Suppose in this pair of autosome, this chromosome might have a gene that is getting affected, which is why I can say that, okay, she is a carrier. Both the parents, this is a female parent, she is a carrier. Unaffected carriers are there, right? Similarly, over here, suppose this is a male. Suppose this is a male and he also have a gene which is being affected, okay, in thalassemia. It's an autosomal recessive disorder. So you know it's recessive. So recessive disorders are only going to affect when both of these chromosomes are going to come together, right? So let's see, when will it come together? This and this comes together, you get something like this. It is unaffected, they are totally fine. This and this comes together, my dear children, you get a condition like this where one is affected but one is still fine. So it's heterozygous, still it cannot express because there is one to overshadow the effect. Again, when this and this comes together, you get one normal and one that is affected. Again, this is also heterozygous. It is not going to show the effect. Now, this and this comes together, my dear children. Both of them are affected, and this is where that particular child, okay, this child, whoever has this, would be having thalassemia disease. So, two parents can be carriers, okay, and their child, there are 25%. How 25%? Listen, 25% of a chance to be, for it to be there, right? Almost 50% chance for them to be carriers, and again 25% chance still left for what? For someone to have this disease. Because out of 4 you are calculating, right? Only 1 out of 4, 1 by 4 is what? 25%. So, your answer would be 25% for someone to have the affected child. If you are asked about the carrier, it would be 50%. If you are asked about the non-affected, obviously they are all non-affected. 2 of them are carriers, but all of them are non-affected. Okay? Fine. Now, which of the following assertion about alpha thalassemia is incorrect? You know there is alpha thalassemia and beta thalassemia. Do you know that? Okay, there is alpha thalassemia and beta thalassemia. Alpha thalassemia is linked with the genes which are getting affected, and these genes are responsible for alpha globulin formation. Okay, so alpha globulin is important for hemoglobin, and similarly you need beta globin chain for hemoglobin. Both the alpha and beta comes together to form a hemoglobin molecule. Suppose this alpha globin genes are not being produced enough, my dear children, obviously what will happen? The quantity of hemoglobin being produced will be also reduced. Similarly, beta, if it is not produced, again the quantity is going to be reduced out there. So, which is wrong or incorrect about alpha thalassemia? It involves both HbA1 and HbA2 genes. Yes, both of them. HbA1 is for coding for alpha gene. Similarly, HbA2 is also going to code for alpha globin gene itself. So, they are both involved. Is associated with the mutation or deletion of genes on chromosome number 11. Chromosome number 11 has nothing to do with alpha globin gene, because the genes of alpha globin are not on chromosome number 11, it's on 16th chromosome. So, this statement is what? Wrong. Is associated with the mutation or deletion of genes on the chromosome 16? Yes. It leads to reduction in synthesis of globin? Yes. So, thalassemia is a disease which we call as quantitative disorder, because since the globin chains are not being produced, hemoglobin is also not being produced enough. Sickle cell anemia, on the other hand, it is known as qualitative disorder. Why? Because beta globin chain, in that some mutation is happening, glutamic acid to valine, which is why a proper hemoglobin is not being formed. It's a structural deformity that happens. So, it is a qualitative. This is quantitative. So, your answer would be what? B. And over here, I have both the things. So, alpha thalassemia, chromosome number 16, beta thalassemia is caused because on the chromosome number 11, there is HBB genes. If they are affected, then you have beta the beta thalassemia. If over here any of these genes are affected, then you can be a carrier, or if all of them are affected, then you can be a major patient of alpha thalassemia itself. Remember, alpha on chromosome 16 and beta on chromosome number 11. How do I remember it? Is that basically B, okay. B usually looks like the half part of B is like 6, right? So, I associate like this is not with this, okay, it's the wrong way. This time it is with A, alpha. I know it's a silly way to remember, but that's how I remember. Yes. Okay. The fact that every gamete formed has either of the alleles for a trait follows that whenever a gamete is being formed, it will at least have one allele. For example, if I take this capital R, small r. So, whenever a gamete is formed, you write like this, no, capital R or small r, right, or capital T and small t. It will definitely have. Why do you write it? What is the concept behind it? Concept of unit factors, law of dominance, law of segregation, law of independent assortment. Independent assortment works well when you have two genes, one gene this and other genes. Over here, you're just talking about law of segregation. Whenever you have pair of alleles, one of them, both of them are going to segregate, and one of them are going to each gamete. So, this is law of independent, this is what? This is law of segregation. Whenever you are forming a gametes, a pair would be segregating. Okay, so C is your answer. Now, next thing. Which of the blood groups is not possible in the child born to parents both having AB blood group? So, both the parents are AB blood group. What is the possibility that the child would not never have? What blood group would the child never have? O blood group. I can just directly say it is what? O. Why? Because both of them have AB. If you do this A blood group, you get A blood group. If you do this, you get AB again. If you do this, you get AB again. If you do this, you get BB again. There is no chance for O blood group to happen. So, your answer would be D. Okay. Okay. This comes under codominance and multiple allelism. Okay, A and B both are codominant. It is also example of multiple allelism. Why? Because one gene has multiple alleles, IA, IB and IO, or you also write it at small I. Okay, multiple alleles. Now going ahead, 25th question. A child having mental retardation and odor in urine is likely to be suffering from. Mental retardation is there and there is odor in the urine. Which is this disease? Sickle cell anemia, thalassemia, muscular dystrophy, phenylketonuria. Sickle cell anemia, you know, the red blood cells become sickle shaped and there is problem with the transportation of oxygen. Thalassemia is again a disorder which is messing up around the hemoglobin itself. Okay. Phenylketonuria is a disease where what is happening? Phenylalanine hydroxylase enzyme is not being produced because the gene coding for it is getting damaged. Now phenylalanine, which is there in our body, it cannot get converted to tyrosine, which is why this guy gets accumulated in our body. And when phenylalanine gets accumulated in a body, it takes up different shape. What do you call? Not shapes, different. It forms up different things. For example, phenyl pyruvic acid. When that is there, it can cause mental retardation and it can also give that mousy or what do you call a very bad odor in the urine. Okay, because it is excreted through that. So your answer would be phenylketonuria. Phenylketonuria is an example of autosomal recessive disorder and also an example of pleiotropy. Pleiotropy means one gene causing multiple, what do you call it, responsible for multiple traits. Okay, I hope you remember that. Now, different types of blood groups are possible in humans. It is possible because of. Different type of blood groups are possible in humans. It is possible because of. Why it is possible? There is O blood group, there is A blood group, there is AB blood group, there is B blood group. Why is it possible? Because it has multiple alleles. There are multiple alleles, which is why more alleles are there, more possibilities are there. Not polygenic inheritance. Polygenic means many genes are there responsible for one particular trait. For example, your skin color, your height. That is why we all have different skin color, because so many genes are responsible for that. Pleiotropy, as I told you, one gene responsible for multiple traits. For example, because phenylalanine hydroxylase is not being produced, conversion is not happening, tyrosine is not being formed. And on the other hand, if tyrosine is not being formed, melanin is not being formed, which is why the people who would have this would also have reduced pigmentation in their skin and the hair. And apart from that, since this conversion is not happening, this is getting accumulated and causing mental retardation as well. So one gene having multiple effects, that is pleiotropy. Multiple allelism is the answer because you have so many things which comes together in different combination, which is why you have different possible blood groups. It's not example of incomplete dominance, it can be an example of codominance. Okay, next. Which of the following cannot be the test cross ratio? 1:1, 1:2:1, 3:1, both B and C. What do you mean by test cross? Ma'am, there is a plant, a tall plant. Tall is anyways dominant, but I do not know whether it's tall like this or it is tall like this. So what do? I cross it with a recessive parent. I cross it with a recessive parent. Now when I cross this with capital T and when I cross this with capital T small t, I find two things. Okay, so if I cross this with this, I get capital T small t, this with this again capital T small t, this with this again capital small t, again capital small t. All of them would be what? Tall. So the ratio is just 1. Over here the ratio would be just 1. Otherwise the other thing that can happen is this. Okay, suppose I will write it like this. This and this goes, you get capital T small t. This and this goes, you get capital T small t. This and this goes, you get small t small t. This and this goes, you get small t small t. So two of them are tall, two of them are what? Dwarf. So 2:2, that is 1:1 ratio is being found over here. So either 1 or 1:1 ratio, but not 1:2:1 or not 3:1. It's wrong. So in test cross means you're crossing with the recessive parent so that you get to understand. If all of them are tall, you get to understand that this tall plant which is there, it has a genotype like this. If all of them are not tall, you get to understand that it has a genotype like this. Okay, that is what you mean by test cross. So both B and C would be your answer. It cannot be a test cross ratio. Okay, next. A patient goes to the doctor worried that a simple cut is taking unusually long time to clot. The doctor identified the patient's condition as, if there is a simple cut and it's taking too much time to clot, that means clotting enzymes probably might not be produced. There might be some mutation in that. Which is that disease? It is hemophilia. Queen Victoria had this disease. Okay, she didn't have the disease, she was a carrier of the disease. Okay, hemophilia. Fine. Now 29th is an example of Mendelian disorders, my dear children. Now the 29th question, which of the following is not possible in a marriage between a carrier woman and a normal visioned man? Normal visioned man looks like this. Carrier woman, my dear children, would be looking like this. You're talking about color blindness, so you know it's sex-linked disorder. What is not possible? Let's see. This and this comes together, a normal female is being formed. This and this comes together, XY, a normal male can also be formed. This and this comes together, a carrier female can be formed. This and this comes together, a color blind male can be formed. Color blind male can be formed. Now let's see. Color blind boys, it's possible. Okay. Carrier females also possible. Colorblind girls, do you see colorblind? This is normal female, this is what? Carrier female, this is carrier, okay, not colorblind. Carrier female, do you see colorblind females? No. So this is something that is not possible. Normal boys and girls, yes, this is normal female, normal male, that is also possible. This is something that is not possible. I hope you got it. Okay, clear? Chalo. Always remember, you have to make a list of, I'll try to give it in the one shot itself, okay. So I'm recording this first and then I'll be recording that. So in the one shot, I'll be trying to give you everything as in a chart so that you can follow it very easily, all the diseases. Okay. Now homework question, the 30th question is a homework question. Tall structure, okay, or stature, underdeveloped testis and gynecomastia are examples or symptoms of what? Gynecomastia means formation of breast. So do you find in Down syndrome, do you find in Turner syndrome, Klinefelter syndrome, or both both A and C? Whatever the answer is, put it down in the comment section. This would be your homework number 3, right? 3, I think so, 3. Yes. Put it down in the comment section. And also, do let me know, was this video useful for you? Did it clear up some concepts? Did you find new questions out there? I'll be really happy to know that. And my dear children, keep working hard. You are giving your best. Wishing you all the best. Lots and lots of love. Bye-bye. Take care. And do not forget to hit the subscribe button if at all you have not done it yet. Bye-bye.